Okay. So hello everybody, my name is Bridlin BARCKMANN. I'm a bioinformatician in the lab of Yvonne Sterkers. And I'm presenting you today the work that we did on the topography of DNA replication origins in Trypanosoma brucei. So this is a project that is supported by Slavica Stanotic in our lab.
Sorry, there's.
I will use the key.
Yeah. Oh, that's good.
No problem. So Trypanosoma brucei is the agent of human African trypanosomiasis, also known as the sleeping sickness. It has two subspecies that are producing the malady, and it's transmitted by the tsetse flies. It is a disease that is fatal if it's not treated, and the geographical distribution is in the sub-Saharan areas in Africa. So I present you here a very simplified scheme of the life cycle of Trypanosoma brucei. So we have two phases. One is in the insect host, the tsetse fly, and the other phase is in the mammalian host. And we use in our experiment a stage that is the procyclic form, which is present normally in the insect host, and a stage which is the bloodstream form, which is normally present in the mammalian host. So DNA replication is the process where the complete genome is once copied before cell division, and this takes place at distinct and multiple sites in the genome that are known as origins of DNA replication. And the defining features of these sites are not very well understood. So there are some elements that have been found to be associated with DNA replication origins in eukaryotes. And these are genetic elements like primary DNA sequences and structures. There's one consensus motif that has been found in budding yeast, the Rs element, but this is the only consensus sequence that has been found so far. All other species don't have a consensus. There are consensus motifs like AT-rich structures, GC screws, or CTPG islands. There are also secondary structures involved. There can be G4 structures, as is depicted here, cruciform structures, and triplex DNA structures. There are also epigenetic elements that are involved or can be involved, like histone modifications or the nucleosome density. Also, origins can be organized in 3D structures within the nucleus, where the origins are then grouped into replication foci within the nucleus. So the aim of our study was to map the DNA replication origins of Trypanosoma brucei with a high resolution and to analyze the structure of the origins. So as I said, we used two life cycle stages, the procyclic form, PCF, and the bloodstream form, BSF, to do this experiment. So one way how you can map origins is by doing SNS-seq. And this is SNS are the short nascent strands that are synthesized in the replication bubble early on. So these are single strand DNA fibers that are primed with an RNA primer. And you have two SNS per replication bubbles oriented in opposite directions. So SNS-seq is isolating these SNS and afterwards sequencing them. So I have a more detailed protocol or description of the protocol here. Here again, you have a scheme of the replication bubble with the two SNS fibers primed with a little RNA primer. So you do first and DNA isolation, and the SNS fibers are at a very, very low percentage within even heavily replicating cells. So you can, it's estimated that you have 0.05% of SNS in the of the total DNA count after DNA isolation. So to isolate the SNS, you have two steps. There's a first step is a size selection, where we select with the sucrose gradient fragments between 0.5 and 2.5 kb. And afterwards, you have an enzymatic purification step where lambda exonuclease is used. And this is a DNA nuclease that cannot digest RNA, so all the SNS fibers are protected by the little RNA primer that they carry, but all the degradation fragments from the genome are digested. So we use from the same extract a control where you use half of the sample and treat it with RNase before the lambda exonuclease treatment, which will remove the little RNA primer, so all the DNA is susceptible to the digestion by the lambda nuclease. So this gives us background control. So we have an SNS enriched sample and an SNS depleted control. So we prepared then from these samples stranded sequencing libraries. And here we made a decision that is different from the normal conventional SNS sequencing protocols, which are normally not done with a stranded sequencing library. So stranded sequencing is a library preparation where you retain the information of the strandedness of the origin strand of the molecule, of the DNA fiber. So here you have an example of, normally it's used in RNA-seq experiments. Here you have an RNA-seq experiment scheme. So you have unstranded library aligned to genes. One of the genes is written in the plus direction and one of the genes is read in the minus direction. In an unstranded library, you cannot differentiate between the directions of the reads. When you have a stranded library, you have the information that this gene or this mRNA was written from the plus strand or from the minus strand. So how can we use this additional information for the identification of origins? So when we look at the orientation bubble, it is oriented. So we have a fiber that is generated from the plus strand and we have a fiber that is generated from the minus strand. So when we can use the stranded information about from which strand the SNS was polymerized and map the origins finer. So I will explain this in a bit more detail on a bit later. So we were sequencing 3 biological replicates from BSF and PCF cell lines from Trypanosoma brucei, from unsynchronized cells with their corresponding controls. We did paired end sequencing, 150 nucleotide reads on a NovaSeq sequencer at the MGX platform here in Montpellier. So here's the overview of the analysis pipeline. So starting with the raw sequencing reads, we have first basic quality control and trimming, adapter trimming and quality trimming step. We map the reads then to the reference genome with BROUTIN. Duplicates are removed with Picard. And we do then, after the mapping, the separation of the reads into reads that are originating from the plus and the minus strand. And we then use these reads to do peak calling like you would do for example, the ChIP-seq experiment, separated for the reads originating from the plus and the minus strand. We then apply a filtering that I will explain a bit more in detail. So there are 2 steps that I will explain a bit more in detail: the strand separation of the reads and the peak filtering. Just very briefly to explain how you can separate the strands, the reads from their strand origin when you have a stranded sequencing library. So depending on the library preparation protocol that you use, you have you can define the reads or the read pairs that are coming from the plus or the minus read on their orientation. They're either the first read is on the plus strand or the first read is on the minus from the minus strand. So the first read is forward and the second would be reversed. So knowing the orientation of the first read, you can separate your mapped reads into reads originating from the plus or from the minus strand. So after the separating, as I told you already, we did the peak calling on the separated BAM files. So so after the peak calling, we have the peaks and we know whether the peaks are originating from the minus strand, originating from reads from SNS coming from the minus strand or the plus strand. So the minus strand SNS are depicted in pink, or the minus strand peaks are depicted in pink, and the plus strand peaks are depicted in blue. So and now we are applying peak filtering with 3 criteria that we assume from the topology of the replication bubble. So we know the orientation of the DNA replication bubble, and to filter for the orientation, we need peak pairs that are in the minus+ orientation. We need the peaks to be in relative close proximity, so we applied a maximum distance of 0.5 kb, and we do not want the peaks to completely overlap, so we have a maximum overlap of 50% of the peaks. So this would be example of peak pairs that we retain, and here example of peaks that we filter out. So this would be peaks that are completely overlapping, peaks that are in the wrong orientation, plus before minus, and peaks that don't have a peak pair partner. So to summarize this again a bit, so we know which reads were generated from plus or minus strand SNS. Therefore, we know the which peaks are plus and minus SNS peaks. We filter the peaks accordingly. We only use peak pairs that are in a minus+ orientation, and then we define the origin in the center between these 2 peaks. So this is a little overview how this looks like when you look at in the genome browser. So this is a part of the core chromosome 1, and here you have the coverage, the read coverage in minus in pink and blue and plus in blue, and the origins that we mapped for the 3 PCF and BSF replicates. Here you have the total PCF origins, the total BSF origins, and the origins all merged. So we mapped in total 125 origins in Trypanosoma brucei. And we then started, after mapping the origins, we started to characterize them. The first thing we looked at was where they were located within the genome, and we found that the vast majority of these origins are actually located in the intergenic regions. So here you have heat maps where the origins are plotted around gene sequences sorted by size, and you see that the gene sequences are actually, or the gene regions are actually devoid of origins. This is a shuffled control, which is composed of regions that have the same size, number, and chromosome location as the origins. We then looked for the nucleotide composition around the origins. So we calculated the percentage of nucleotide within a 20 base pair window, and we plotted this around the origin centered in the middle. And you see that we have a strong increase of A nucleotides upstream of the origin center, and a strong increase of T nucleotides downstream of the origin center. So we have a very specific structure of nucleotide composition. We also see a slight increase of G in front of the origins and of C's downstream of the origins. We do not see this at all in the shuffled controls. So we have this situation where we have poly A enriched regions, point symmetric on the plus stream upstream of the center, and then the minus stream downstream of the center, and vice versa for poly T regions. So the next thing we looked at was G4 structures surrounding the origin centers. So we were lucky to have a data set of experimentally determined G4 structures. So G4 structures are elements in the DNA composed of groups of Gs that can form these secondary structure in the DNA. They are involved in a lot of I don't know a lot of different processes. That's what I wanted to say in the cell and the nucleus, like also they're involved in transcription and things like this. And they are also published to have implication in replication. They are found in a lot of eukaryotic cells in the vicinity of origins. So when we look at what we found also a very specific localization for these G4 structures. We found them in the plus strand upstream of the origins and on the minor stream, directly downstream of the origins. So we find that 74% of the origins are actually flanked by at least one G4 structure. And when we look at the localization in comparison with the A-enriched regions, so here in the dotted line is the poly-A enrichment, in blue for the minus strand, and in red for the in blue for the plus strand, and in red for the minus strand. And the line is the G4 signal. You see that they are completely in the same position, meaning that the G4 structures are actually embedded within the poly-A enriched regions. So this was something that has not been seen like this before. G4 structures have been found to be associated with origins and also AT-rich regions, but that the G4 structures are completely embedded in these enriched regions has not been seen like this. So we also looked at the nucleosome occupancy around the origin centers. Here also we were lucky to have an MNase-seq data set available. And we see that we have a depletion of nucleosomes directly at the center of the origins, and then 2 shoulders of nucleosome enrichment next to the center of the origins. We also looked for the association of R loops and around our origins. So R loops are structures where you have an RNA fiber hybridizing with one strand of the DNA double strand, and the other strand is displaced. So these structures can be found throughout the genome, and they're very often associated with transcription. There are also some hints that they could be associated with origins. So when we do the overlap with our origins, we see that we have a strong association of these R loops with our origins. So we have 90% of our origins overlapping with R loops. And when you look a bit closer with this little double summit or double tip at the summit, and we are hypothesizing that these could be actually the enrichment of RNA primers that are the beginning of the SNS fibers within the replication bubble. So with the stranded SNS-seq, we were able to propose a model for the landscape of the origins in Trypanosoma brucei. So we have a very organized situation around the origins. We have poly-A enriched regions upstream, poly-T enriched regions downstream, and within the poly-A enriched regions, we have the G4 structures upstream and downstream on the minus strand. The center of the origin is nucleosome depleted. We have this enrichment of nucleosomes next to the center of the origins. And we also have this strong association of R loops over the origins. So now I would like to show you in one slide why the stranded SNS-seq sequencing was increasing our resolution quite a bit in comparison to the conventional sequencing where no stranded sequencing is used. So here I show you again the scheme of the replication bubble and the peaks that are resulting from the SNS, isolated SNS, coming from either the plus or the minus strand. We have this information because we used the stranded sequencing. If we would have used a normal sequencing, this peak we could not have said that this peak is coming from the plus or the minus strand. They would have all the same quality for us. So we would have placed the origin, and this was conventionally done in the SNS sequencing, at the summit of these peaks. So because we have the orientation of the of the peaks, we can apply first the filtering. That means we can filter out a lot of false positive peaks. And also we can afterwards place the origin in the center between these two peaks, which is way more precise. So I show you here the analysis I showed you before over our origins, the distribution of nucleotides. And when I use from our experiments, all peaks that have been called and do the exact same analysis, I do not see this. So this information is hidden between first all the false positive peaks that you have in this experiment. that we can filter out, and also because we are not precise enough to place the origin in the middle of these two peaks. So with the stranded SNS-seq, we have a much higher precision in origin mapping than with the conventional SNS-seq technique. So I would like to thank the team of Yvonne Sterkas, especially Slavica Tonic, who was carrying this project. I thank our collaborators, Peter Monsignor in Belgium, who helped with some of the analysis, the sequencing platform MGX in Montpellier, and I did all the analysis on this project on the Roscoff platform cluster. And I thank you for your attention.
Thank you very much. Has anyone got any question or on the chat?
Thank you. It was very interesting. I had a question. Do you define with MGX the insert size of the between the forward and the reverse?
I mean, this is defined by the DNA fragments that we're sequencing, which is first defined by which is defined in the end by the sonication that we do in the library prep.
But is there a specific size because your fragments are so small? Or is it the...
So do you mean the peaks?
No, the insert size between the forward and the reverse? Like, do you sequence them?
No, they're relatively short. Like what the sonication was aiming for, what size of... I did not do the library prep, so I am not 300 nucleotides. So they're close together.
So they never overlap?
Honestly, I didn't check that.
Okay, thank you.
No.
Hi, Bridlin. Just a question. Do you also find, or there was found the G4 structure they were associated also in Leishmania, or just something that is specific to brucei?
In the Leishmania, I don't remember if they checked the G4 in the... I don't think they checked for the G4. Do you remember, Slavica?
G4 and origin in Leishmania, they are never checked, but we know from the G4 sequencing that there is much more G4 structures in Leishmania than in Trypanosoma. There is something like 3-4 more G4 structures sequenced in Leishmania than in Trypanosoma, which is kind of logic because the genome is more GC rich in Leishmania compared to Trypanosoma.
It's just a question because in that case, the G4 structure, I understand that could be more related to having a...
I don't understand you very well.
The G4 structure, what do you think about the interest to have, what would the...
So it's not really known what function they have at the origins. There's hypothesis that they could cause the complex that is building to slow down the process a bit before actually licensing the origins. But there's no real knowledge on what they're actually doing.
Because it's more associated, possible, the G4 more with the regulation of transcription
also, when you have the
or
G4,
not?
you don't have the double helix, so it's also kind of maybe opening the DNA helix, which is also necessary for the replication. That would open the replication always profits from structures that are loosening up the double strand.
Okay, well, thank you very much. Donc maintenant, vous allez avoir un talk commun des 3 GT 1.5, GT équité diversité et le groupe G.
Donc, la direction, Frédéric, Sylvie et moi, on voulait vous faire un petit point pour le contexte de cette présentation. Donc, en fait, en début d'année, on a mis à jour le règlement intérieur du laboratoire. Donc, pour rappel, il est disponible sur Synology dans l'instance MIVEGEC pour tout le monde. Donc, ça regroupe tout ce qui est réglementaire au niveau du cadre du travail sur les horaires, les accès, etc. Et on a décidé cette année de rajouter un document complémentaire sur ce qu'on a appelé l'organisation et le fonctionnement interne de l'unité. Donc, ça permet de décrire un peu tout ce qui a été mis en place pour améliorer, favoriser et être au mieux dans les conditions de travail du labo. Donc, ce document, il a été rédigé avec l'aide du CUCL et donc de ces 3 GT et transmis au COPIL. Et il est organisé. Il faut cliquer. Je clique. Organisé en 4 chapitres, dont les 3 premiers ici qui sont très succincts, c'est vraiment moins d'1 page par chapitre. Donc le premier, ça rappelle les thèmes et l'organisation, les thèmes scientifiques et l'organisation en 5 DS de l'unité. Le deuxième rappelle la gouvernance, donc avec les 3 instances actuelles et leur interaction pour la prise de décision. La troisième, c'est tout ce qui a été mis en place en termes de groupes, de fonctions, de procédures, etc. Pour le fonctionnement interne. Et le 4e chapitre, dont vous allez entendre parler maintenant avec les 3 GT, c'est ciblé, donc c'est nouveau, et c'est ciblé sur l'éco-responsabilité de nos activités de recherche et la responsabilité sociale au sein de l'unité. Donc, on a 3 GT qui existent pour certains depuis de nombreuses années, qui ont fait beaucoup de sensibilisation, d'information et des propositions. Et donc, on s'est dit que là, il était temps de passer aussi à l'action. Et c'est ce qu'ils vont vous présenter, chacun avec leur spécificité. Donc, vous allez avoir une liste d'actions concrètes qui vont vous être présentées en termes de règles et recommandations. Donc, les termes ont été bien discutés. L'idée des règles, c'est qu'on voudrait mettre en place ça, on va mettre en place ça à l'échelle de l'unité pour que ça devienne un fonctionnement de routine, le fonctionnement de base pour ces actions-là. Et les recommandations, l'objectif est le même, sauf que ça part avec un choix individuel, si on parle des missions, par exemple. Donc, on souhaite évidemment un engagement le plus complet de l'unité. On vous invite à suivre ces recommandations, mais il était important de faire la distinction suivant les actions qu'on allait mettre en place. Donc voilà, on laisse les groupes présenter et surtout, là, on en profite pour remercier toutes les personnes qui participent à ces groupes. Ça prend du temps, mais c'est hyper important pour le collectif. Et on n'oublie pas aussi que les groupes restent ouverts. Donc, si des nouvelles personnes volontaires veulent s'y investir, je pense qu'elles sont les bienvenues.
Hello. Then last time we have focused on the proposal from the Greenstreaming project that has not been really highly approved by you. And today we will focus on the proposal that has been validated by the CUCL because it has been discussed in the CUCL since then. Actually, they have validated all the proposals that had a good score and also some of the proposals that had a bad score, but they slightly modified it to make it acceptable. Then I'm going to present you those proposals that are now rules or recommendation. But I go quite quickly because it's a bit long and we have no time for all this. But after validation by the COPIL, which is the next step, we will make a written report to share all the results of this Greenstreaming project. Then they are divided, as said just before, into rules or recommendation. Rule is something called compulsory. Recommendation is something that really you should do it also, actually. Well, the title is not visible, but anyway, as for the purchase, now we will buy recycled or recyclable paper and also all the everything we can buy as recycled or recyclable. Also made in France products, vegetable ink when possible, and rechargeable devices and batteries. As for numerics, we won't use any electronic signature with pictures in email, and we will share a calendar to reduce the number of emails to share webinars or seminar or other events. As recommendation, it is advised not to use attached files over 200 kilooctets in mailing lists. And also, we are invited to use listening platforms instead of video platforms to listen to music in the lab. We shouldn't buy a monitor if we have already one which is working, and we shouldn't buy any computer if ours is less than 5 years old. At the bench, we will use reusable socks instead of disposable ones, and we won't autoclave systematically everything, but only equipment that really needs to be sterile. And as a recommendation, we will ask for a shared laundry position for all the labs in the center here. As for the cold storage, actually, it's already done. We have a schedule to sort and tidy fridge and freezers. As a recommendation, we should always properly identify the contents of the cold storage. And it's advised to really take time to sort and clean the contents of the fridge and freezer instead of buying new device when it's full. As for the travels, now we will have to take the train instead of a plane when the equivalent journey is less than 5 hours. People are also invited to do visio instead of traveling for meetings. As recommendations, it is advised to take the train instead of the plane when the travel is less than 8 hours. And it's also advised to avoid to take plane if you are going to stay inside less than 3 days. And it's also advised to take the train instead of the plane for pre-routing to reach an airport. As for drinks and meals, now vegetarian meal will be the default choice during seminar or trainings. It is advised to use the cups from the MIVEGEC kit instead of disposable ones. And it is advised to, well, it's advised to choose a meal provider in the list that we have already shared with you that you can find in the intranet site. It is recommended not to stop buying non-recyclable or non-compostable coffee capsules, and to avoid buying new devices for coffee or tea. As for the evaluation process, people leading projects for over 1 year, as well as people organizing seminar, are invited to assess the carbon impact of it with the tools that we are about to create. And to finish, as for the energy, now the maximum temperature set in the labs will be 20 degrees in winter, and the minimum one will be 25 degrees in summer. And that's it. Thank you.
Okay, so now I will talk in the name of the G. So we also have some recommendations. First, to facilitate work-life and talking about right to disconnection. So to avoid working during the weekend, for example. So to do so, it's important to be careful with the email that you send. We each have our own rhythm to work, but our rhythm can impact others. And this we need to take it in consideration. People, if they don't want, don't open their emails, but it can be easier for some people than others, considering personal state or hierarchical relationship. So instead of putting the responsibility of the one receiving the email, it's important to also take the responsibility of our actions. So when you send an email, like you can always think, do I need to send the email right now? Is it important that it needs to be now, or can it wait for tomorrow? The worst is like the email like on a Friday evening, and like you keep the message all the weekend. So a great tool to avoid that is email. You can make a draft that you will send later, but you can always forget about this draft and by the end not sending your email. So what you can do is like you write your email, but instead of sending it right away, you can program it to send it later. So I made a little how to say, protocol, yeah, to show you how to do so. So when you write your email, then you can click on send it on the arrow beside sending, and then like you click on sending later, and then ta-da, you can choose the time, the date, the time, even the timeline, if you're on a different timeline. So, and you can be sure that your email will be sent and received at a proper hour for the one that will receive it. Another way, it's also to communicate by email as much as possible and avoid instantaneous communication tool. And like you have to take in consideration like the impact of these tools, because like most of the time, like you can check when the person saw the message. And so when the message is seen, like it's like, why is not answering? So it can be easier to in some aspect, but also put a bit more of pressure. So it's better to do it by email. Another topic, it's like the hierarchical relationship, how to improve them. So I hope we all agree that slavery is over. And so to have a great hierarchical relationship, it's to discuss, because sometimes like we all think that we are on the same page. And at one point, there is some conflict because by the end, we are not on the same page. So a good way to avoid that is to discuss at the beginning of the contract. Can be like a PhD contract, can be an internship contract, can be any CDD contract. So you discuss to clarify time schedule, to define working days, to define communication methods and how frequent and like everything. So the objectives. So we have written what both sides expect. Also, it's to establish the workload, and it's important for the PI to be conscious of the real workload, and can be a bit tricky for PI that might not be in laboratory anymore. So I think it's important to keep in mind how long does a PCR take, or how long does an analyze take, to be sure to not ask too much. And always always be sure to have a clear and respectful work environment, and this can be like by discussion, by a good communication. And to preserve the well-being of everyone inside the unit, because well-being, it's like the top of the house to keep everything together and to have someone that is motivated to come to work, do an efficient and good work. Well-being is like the key. So it's important to stop rumor, like when you listen to something, like instead of like just, oh, I heard something, I will tell you. No, like if there is some rumor, like stop spreading it, and instead like go to see the concerned person or like people that will take action to solve the problem. It's important also to break the false idea about the benefit of stress. For example, stress is part of the job, or you need to work night and days to get a good thesis, or if you are stressed, it's always the fault of the PI. No, stop. Okay, there is many reasons to be stressed. Stress might not be good. In many cases, it's not good. So let's stop it. And you can always like self-evaluate your work condition. And for example, you have this bioinformatic that you can all print and, for example, put at the door of your office to be sure that your work environment is always respectful. And well, you can, so you have the link that you can also find in the website and in the intranet and the QR code to have more info about it. But I think it's also good to know ourselves where we are placed in our work environment. I see some QR code scanning. It's okay. It's in the intranet if needed. So this is the actual G team, and well, we are always open for new people joining the team, so don't hesitate, and thank you.
Okay, so hello everyone. So I will speak. Yep, thank you. So yeah, quickly, I will speak in the name of the group about equity and diversity. Just a quick reminder that we created this group to inform, exchange, and raise awareness on the issue about equity, diversity, inclusion, and accessibility to perform a good science altogether. And basically, our two major actions are raising awareness through information and sharing and events, but also encouraging in terms from background that are usually far from science. And so today, in terms of rules and recommendation, we not working. Yes, I'm clicking. Okay, so les flèches marchent plus. So in terms of rule, we just perform one proposal, which is now to implement inclusive communication within the unit as a rule for systematical official exchange of documents, which includes a website, job profiles, quick announcement, newsletter kind of thing. And of course, we provide tools to do so. So feel free to ask for more information. And just a quick reminder that is by the law requires that we use that type of inclusive communication in job profiles. So this is the only one that we did for this time, but it's still a work in progress. And especially we are still eager of new idea about themes or events. So please feel free to contact us to make that suggestion, or even if you want to start an initiative on yourself, we can help you. And also we can help to gather information about themes. Also feel free to join our meetings. We are a small group, but with highly motivated people that want to help our environment to be better. So feel free to use the mailing list. And also, this is a good time to say that the group coordinators have changed. It's now Sylvain Pinault, which apologize not to be here today, and myself that are now coordinators. So also feel free to contact us directly. And also very soon there will be a file, a directory on Synology to find all the information that you want about this subject. So feel free to try one meeting and then see if it's for you. Thank you.
Hi everybody. My name is Johannes. I work in Artus Lab since January, so I'm pretty new to the IRD. Today I'm going to present you a project that we have in the lab kind of for a long time, which is about spatial transcriptomics in pregnancy-associated malaria. And I also give you a little bit of an introduction to spatial transcriptomics. So what is spatial transcriptomics? It's, well, a collection of different methods that utilize different methods of measuring RNA quantities, either by imaging methods, microdissection, ISS, or what we normally do, or which is also like the trend nowadays, is sequencing-based methods. So how does it work? It's essentially pretty simple. You have something like an array, which is similar to the array that you had, back in the days to measure gene expression, or what you have in Illumina sequencing, for example, on flow cells. So we have mobilized oligo-DT primers that have a special barcode. So that spatial barcode is essentially encoding the position on this array, and with the poly DT primer or oligo-DT primer, you capture the mRNA that is polyadenylated. There are some other methods that do not use the system, but use probes. They are more, well, they are better for like FFPE samples, for example, because in FFPE samples, this doesn't really work. But very often it essentially follows the same recipe, which is you do some reverse transcription on the chip, including in this case, so this is 10x Genomics, for example, the vision technology with template switching at the end. You do second strand synthesis, and then you elute everything and you do some PCR. And for the evolution, this is a technique that is like, the evolution is absolutely crazy in the last, I would say 10 years now, yes. So this is from a paper that was just, no, not published, but put on bioRxiv, from a new method called SeqScope X. And you have a huge reduction in the increase actually in the resolution. So you capture smaller and smaller areas, and the distance between the areas that you capture gets bigger and bigger. So we are essentially now at a subcellular level concerning the spatial information that we get. So why do we care about spatial things? The reason is because cell-to-cell interactions matter a lot. And essentially, what you need to always keep in mind is that if a cell is in a certain environment, that can be, for example, in a disease environment like the tumor niche, so the microenvironment for tumors, or tumor heterogeneity is a big topic. We need to understand which cells are where, essentially, and who are the neighbors. The same is true for, let's say, for example, for tissue development or general like embryonic development. It's also a very, very interesting topic. And with spatial transcriptomics, what we get is we can not only identify cell types, or at least have proportions of cell types if you are not on a single cellular level. We can also see where they are in the spatial, well, in the space. We can analyze cell-to-cell interactions. We can see spatial expression patterns that might be more like gradient-wise rather than actual cluster-wise. And depending on the method, we can also look at subcellular things. For the bioinformatic analysis, you're spoiled for choices as always. But essentially it follows mainly, or right now, mainly like single-cell RNA sequencing analysis tools are used. I think most people that have something to do with single-cell RNA sequencing know the Seurat package, or Seurat package, I don't know how to pronounce it actually, or the equivalent would be ScamPy in Python. That is used quite a lot, I would say. There are also other packages. I would like to mention Sparta here. This is one package that is more or less like a whole pipeline from the beginning to the end. That is actually quite, I wouldn't say famous, but quite known. Right now, there's actually Sparta 2, so this paper is way out of date because it's almost 3 years old. But there are also like, many others. So you have plenty of choices, which is sometimes good and sometimes not so good. So for us, what are we interested in? We are pretty much interested in parasite-host interactions. And while this applies to many different fields, for example, this can also apply to plant science as a very big outlier now. This, for us, what we are interested in is malaria, so Plasmodium. And in humans, there's one thing that happens during, or can happen during pregnancy, which is when you're infected as a woman that is pregnant, infected erythrocytes can sequester within the placental tissue. So what does that mean? That essentially means that you have a disruption of the placental formation and the structure. So what that can lead to at the end of the day is, in the worst case scenario, prenatal death or maternal death even. You very often have something called, or similar symptoms like in preeclampsia. So this is a condition where you have high blood pressure. So the mother has high blood pressure and liver issues and kidney issues, which then leads to preterm delivery and fetal growth restrictions, or SGA in short. And the severity of that ranges a lot. There's a lot of heterogeneity, so it's not super well understood. And we were actually thinking, so what actually happens on a cellular level here? So what was done way before I joined was that from 4 uninfected women and 6 infected women, the placenta was sampled in Benin. So this was done by Sebastian and Celia. And this is a term placenta, so it was after the birth of the baby. And then one tissue slide was used per placenta to generate 10x Visium library. So these are the old ones where the diameter is not subcellular or single-cellular. So it's like 55 microns in diameter, and between each spot is 100 microns distance. So it's like mini bulk, you can say. So how does that look like? It looks like this, for example. And one thing that we saw here, which is very, very interesting, and that needs to be actually answered, the question is, we have some, in the EVT subclusters, we have, or in the EVT cluster, we have some subclustering in 3 of the patients, or not patients, women, or placentas. So you can see it here, for example, the sample 01cc. So these are all spots that were assigned to be mainly coming from EVTs. So they have a very strong EVT signal. And we have these 2 subclusters identified. So I call the yellow and the blue one. And you can see here very nicely how this essentially follows the histological structure. So you can see this, I'm really sorry, membrane-like structure. I'm really bad at histology. And in the other 2 samples, it looks a bit different, interestingly. So here again, we have yellow and blue. And you can see here in the blue area, we have a higher density of the tissue. And the same is true essentially for the 3rd sample where we have kind of a mixture because, okay, here it's actually orange and blue. We have this orange line that resembles a little bit like this membrane structure that we saw in the first one, but not 100%. So at the moment, we have actually no idea what that means. Or not much idea, let's put it like this. But what is very interesting here, so there's quite some similarities between the three patients when we look at the differentially expressed genes between the two clusters, but there are also quite some differences. What is true for all of them is that the kind of cell type scores that we got from deconvolution are generally lower for extravillous trophoblast within the yellow cluster. So that is the cluster that is actually, it's a bit more like endothelial cells, but it cannot be really much explained by this is a mix of endothelial cells and EVTs. And additionally, we have some signal that might come from actually being infected, which might also sense because we have two infected here and one that we labeled as uninfected, but was actually previously infected. So this is one of the interesting findings that we had. Further on, we combined all the data together and then asked what's actually the difference between uninfected and infected. And for that, we again looked at each, I would say, cell type individually. So again, in the EVTs, for example, we got quite some differentially expressed genes. And when we look at these, some of them make absolutely sense. So these are just, it's just a selection of genes with a fold change higher than 1. We see, for example, overexpression of HLA, which is important for maternal fetal tolerance. And a disruption in the gene expression in both directions here can have major impacts on the pregnancy. We also have FTL overexpressed, which is an iron storage protein. That might make sense or more sense a little bit later. But we also see some things that we didn't expect. For example, TAC3, this is a gene that is normally upregulated in preeclampsia placentas, but in our case, this is downregulated in EVTs. We look at some gene enrichment analysis. We do get some interesting stuff. So, for example, in the upregulation, we have some immune system related reactome pathways, but we also have them in downregulated genes. So what does that mean? So we have some immune genes that are upregulated, some that are downregulated. And interestingly, in the upregulated fraction, we have genes that are associated with ferroptosis. So this is an iron-dependent apoptosis pathway. We have downregulation of some integrin, interferon, interleukin associated genes. And we have quite a lot of genes that are associated with stress, hypoxia, and preeclampsia, and some of them are downregulated and some of them are upregulated. When we then look at the biggest cell type, essentially, or not the biggest cell type, but the cell type that makes the majority of our data set, which are the syncytial trophoblasts. We have 3 different clusters here actually within the cell type, which we don't understand yet. But when we look at the differentially expressed genes between infected and non-infected, again, we see quite some overlap in all these 3 clusters. So this is cluster 0, cluster 1, cluster 2. So what you can see here, for example, ADAM12 is a gene that pops up, I think, in at least 2 of them. Other genes that are overexpressed would be, for example, MALAT1, which is highly involved in the immune response, including preeclampsia, PAPA2, also known to be upregulated in preeclampsia, CRH, whose upregulation is correlated with small embryo size, and ADAM12, which is, as I said, also in preeclampsia. And we have some genes that are downregulated in MAST and SPARC, which are also somehow related to either preeclampsia or different functions within the placenta. So this is all very nice, but this doesn't require spatial information much, except for maybe the subclustering that I showed you in EVTs. So what we were really interested in, what actually happens at the site where there's a sequestered parasite happen. So what we did is, as we got not only human genes, but we got also, or RNA, we got also the Plasmodium RNA. we essentially assigned spots where we were sure that, well, not sure, but where we think that there is a sequestered parasite. And this is how it looks like in the 6 patients, and you can see there's quite some differences. We excluded from further analysis these 2 because essentially everything seems to be somehow a sequestered parasite, which is not really possible. But what we wanted to see then is what happens essentially in the spots around the parasite. So we calculate the Euclidean distance towards these loci, and then performed and then kind of grouped them into affected and non-affected. So the spots that you see here, these are the affected ones. So it means that here we have the sequestered parasite, and around essentially in the direct neighbors are essentially what we call affected. Oops, that was too much. So when we then did differential gene expression between affected and non-affected on the 4 pooled samples, because the amount of spots that we have is very, very low. So to have a bit more power, we pooled the 4. We may only see a signal within syncytiotrophoblasts, which makes sense because that's the only cell type where we have enough spots to have a certain power. And you can see there's quite some differences. When we look at subgene enrichment, all of those reactome pathways in this case are driven by 2 ribosomal protein genes. So we were not really sure if that is a good signal here. But we see some interferon gamma signaling, interferon signaling in the adaptive immune system in the upregulated ones. So this is a bit different than what we saw before. And if we look at the genes, you can see that the expression of the genes, so this is the normalized expression. These are some of the immune-related genes that are differentially expressed in the 4 patients or in the 4 placentas. And you can see the differences are not super huge in the differential gene expression, but there are some. For now, this list of genes does not make a lot of sense, I would say. Except for one, which is not there, doesn't matter. So what we have here is that some of them seem to be somehow related in the recruitment of immune cells, but there are just a few of them, so we're not sure if that is a statistical signal yet or not, like if that is really relevant or if it's just by chance. So in conclusion, when we look at the different cell types, essentially mainly SCTs and EVTs, we have up and down regulation of immune genes. We have possible heterogeneous adaptation per cell type in response to the infection. We have some similarities though, which is very nice, and it makes all sense in the context of preeclampsia. The local responses are really, really hard to analyze at the moment. There are 2 reasons for that: the amount of data that we have, and second of all, we are not 100% sure where the border distance should be. So how far should we go outside of a sequestered parasite to see if there is an effect? So lastly, I wanted to give some future application possibility. So first of all, we will obviously use mainly like the newer techniques with a higher resolution from now on. There's even now Illumina will launch another one, just, you know, to be also in the mix. And one project that we have where this might come very, very handy is avian malaria. So in avian malaria, the big difference between avian and human malaria is that Plasmodium this Plasmodium species can infect endothelial cells and not only erythrocytes and hepatocytes. So you can find them essentially in very different tissues. So from the liver and kidney, but up to even the brain. And it's super diverse. And here though, it would be super interesting to, you know, look what happens essentially as a result of an infected endothelial cell within the tissue. With that, I'm already done. Big thanks to Cécile, who did all the library prep in the lab, and all the spatial stuff. Then Sebastian and Celia, they were essentially collecting the tissue and organizing everything. Quentin did the initial analysis of the data, and obviously Arthur, and thanks to my two co-workers, Sylvain and Iris, Iris is doing the avian malaria. So thank you for listening.
Is there any question?
Thanks very much for a very nice talk, particularly the last bit. No, I'm joking. I work on avian malaria. This is a really silly question, but because affected and infected sound so similar, I got a bit confused with your definition, and I actually would suggest that when you give a talk, you use a different term, because so the affected cells are the cells that are immediately adjacent, like physically touching.
Yes,
because
physically touching. So sorry.
1, 2, 3, 4, 6.
Yeah. So essentially, you have the spot here,
yeah.
Which has the star is the one where we assign there might be a sequestered parasite in the spot.
Yeah.
Then you will have, there is physically on the micro area or the area, there's a an empty space where there are no spatial barcodes. That we don't get in this method. And then the next spot is the one that we took as an affected, so it's influenced by the sequestration. And if we then go, for example, these are not plotted here, but a spot that would be here. So this is actually the spatial dimensions. A spot that would be here would be assigned not affected or not.
Except that you haven't seen very big differences between affected and unaffected. So they're not affected. Your affected cells are not affected. Is that the conclusion?
There's not yet a real conclusion to it. The issue is one issue is how we define it. We are not 100% sure that we should only look at the ones that are exactly next to it. It might also be actually the same way that it could be such a narrow signal or such a narrow influence that the resolution here is not enough, that we dilute the signal by taking tissue parts or cells that are already too far away. But there could be also the reason or something that the influence area here is too small, and we should look at a wider range.
Yeah, thanks for the nice talk. I have two questions, technical. First, so what is the resolution of genes you get per cells as compared to single cell? Like how many genes can you determine per cell with your spatial transcriptomics?
So that depends a bit on the method. I don't have the exact numbers in my head, but for this old school Visium, I would say it's similar to single cell RNA sequencing. However, each spot is more than a single cell. So the efficiency of capturing the RNA is less than in single cell. And in the new methods that have, for example, I think the newest 10X will have 2 microns per spatial barcode. And I think Lumina will have 1 micron even. You will get much less genes. So you are around, I would say, like 200 to 500
genes per cell.
Genes per cell.
Yeah. So your resolution is lower, but you get the spatial information, which is interesting. Is it possible that once you go sideways to the clustering of your cell types, and then determine your cluster cell types, and then come back to your spatial transcriptomics, spatial distribution, and see if you have different cell types distributed on your spatial dimension. Like, would you find some cluster of cells that are more prevalent around your parasites than just taking the spatial information and looking at clustering?
We looked at that. We don't see an enrichment. We thought that, or what we hoped a bit, is that we maybe see like immune cells recruited to these sites. Interestingly, and this is a bit of an issue, I would say, with the size of the data set and the heterogeneity, we only have one patient where we actually have a good signal for immune cells like T cells, macrophages, and stuff like that. So sadly, we don't see any big differences there.
Thanks.
Good.
Just a clarification. These are erythrocyte stages of the parasite.
So it's not, so here, it's not a single parasite, it's a infected erythrocyte that is sequestered with the parasite inside.
Yeah.
Okay, thank you very much.
I got 2 slots for the price of 1, so I will be slightly longer than the others, but I hope to not be too long. So the topics here will be broader. We'll talk about FAIR principle. Refer, that the meaning behind is like reproducibility, refaire in French, and the need for transparent science. It's the idea behind is the open science. So why these 3 topics? I think nowadays is the 3 main pillars that allow and are fundamental and give the good framework to do good science. So we need really the FAIR side, the reproducibility, and the open science together to do good science, good research. So let's focus first on the FAIR. FAIR, it's the acronym for findable, accessible, interoperable, and reusable. And it's about data, and data is everywhere. Actually, everything is data. So even at its experimental side or the data itself, the code is data. When you do analysis, the environment is the data, and the results are data. So the FAIR principle will apply to everything. And the data should not be a mess. It should be well organized, and you shouldn't end up with something that is not handling easily. So to give an example here is how do a well mismanagement, the mismanagement in practice. So maybe I will not have time to go into detail because I have a lot of slides. But some typical problems will be during the analysis at the very beginning here. You have raw data, and we tend to manually edit this raw data. And this is not reproducible by definition. There is also sometimes you use tools to do addition of the data, and you forgot to keep track of what has been used for the parameters, and then it's not reproducible at all. And since at the very beginning, then you have a problem. And something I'm really attached to is when you use a cluster or HPC cluster, usually you put the data there, you are ready to publish, and you are really happy, and you don't think at your data at all anymore. And suddenly you receive a mail from the IT that say, we have soon, we will close your project, and people react and say, I have to save my data somewhere, I didn't think about it. So it has to be planned really early in any project. And unfortunately, finally, it ends up often where the data will just be saved on a hard drive on your desk, and this is something that you should definitely avoid. So what we would like is something really clean, that data, we know where they are, how to retrieve them, and easy to analyze. Is why there is a DMP, the data management planning, that is good and that start at any project that will give you a vision of what you will do, what data you will produce, and so on. And also the FAIR principle that is a guide actually to how to handle your data at any step of your project. Oops, sorry. So this is an explanation of what is the FAIR principle. This is not at all exhaustive, just a few examples for each letter of the acronym. But the most important, I think, is to be findable, a data has to have a persistent identifier, like a DOI, that we know this data as a unique identifier. And this unique identifier should be used to retrieve easily this data from a database. And accessible is different than open. Often people make a mix-up between the two terms. Actually, you make the data as open as possible and as close as necessary. The metadata has to be open to everyone, but the data itself, you could choose to share it with everyone you want. You no need to open everything, even if I think it's better to open as much as possible. For the interoperability side, use open format. This is something that is often forgotten. And for the reusability, there is needs for a license that give a clear information to any users what can be done with this data. It can be reused by another research project and so on. So even following the FAIR principle, at the end, there is plenty of database where you could archive your data. And this is really a jungle. There is plenty of databases that follow the FAIR principle, actually. But they will ask for different metadata or different format of the data, etc. So it could be anyway difficult. So handling data is very, very difficult. There is more and more professional of the data, and we call them data manager or data steward. Unfortunately, we don't have any yet at MIVEGEC. We could hope to have one in the future, but this is kind of job that we see more and more in different departments or in different places. And if you touch sensitive data, like human data, there is an extra layer of complexity, and here there is a DPO, the data protection officer, that would help you to deal with that. So what will be the good way to handle your data? So in a research project, the best would have to have the data and the metadata all deposited in a public repository. The raw data should be in unedited form. And if you had to pre-process this data, if it's edited by a script of software, use a script of software and never do it manually, as I said previously. And if you use script and software, think to share the script you have used or the version of the software and the parameters for sure. Always add a section in your paper to help for the reproduction, and use non-proprietary data in machine readable format. So for bioinformatic processing later on. So use CSV rather than XLS. XLS should be completely forbidden when you do bioinformatics. Or TSV or whatsoever, not necessarily CSV. So actually, there is plenty of tools to help you to deal with data and follow the different principles. And this is not an exhaustive list, but you should learn few of them at least, and they are here to help your life as a researcher and to help you in this to handle this data properly. So why spend time with all of that? So making the data fair is true, is an extra layer of work, but at the end is it's benefits for you at many levels. So the quality of the data itself would be probably better. You will gain in reputation, time efficiency, maybe not at the beginning, but at long term, you will save time actually. You will gain visibility and transparency. Probably you will have a feedback and more collaboration opportunity. And with nice data, you will have with new collaboration, but possibly new project research using this data. So let's talk now about the second pillar, the reproducibility. Maybe it's a longer part. So reproducibility is difficult for the experimental side. So it's not there that we will focus, but reproducibility is more in the analysis itself. So it's about the data, the code, if you have a script, and the environment where you do the analysis. When we talk about reproducibility, I don't know if you see on that corner, but it means with the same code and the same data, this is reproducible, you should get exactly the same result. It is expected if the things are done well, but it's not easy. It's not an easy task. So let's talk about the data first. So to be reproducible, first, the main point is get organized. This is simple, this is common sense, but you have to think and be really strict on the way you work. So all the data is just file and folder. And this is maybe simple, but you have to think at the beginning to be consistent when you name things that it's meaningful for you and for others also, not only for you. So in that example, if you have that on your computer, you should definitely avoid, because we don't know which file is the last one, what is this extension, what tool I need to use to open this stuff. No, definitely avoid that. So a few tips here. So what you should do is, including version, if applicable, use format extension that have a meaning. Use the standard like for years. Always add a README in a folder to explain what has been the name convention or what is the directory structure and abbreviation you could have used in that folder and so on. Avoid using space in file names. I encounter that quite often still nowadays. Always use underscore or dash. It's not to annoy you, it's just because when you need to process it bioinformatically or informatically later, the space are problematic. So avoid special characters in file names and long names, then it could be problematic. Try to be short and meaningful. So the directory structure itself, it's good to have distinct project folders, so one folder per project, and use relevant subdirectories. And this project folder should contain everything, all the file needed to go from the raw data to the final results. So this is an example of a project organization. You could have your own. It just has to be meaningful for you and that understandable for the others. So in that case, here we have the code that could put in one folder specific, the data in one other where we never edit the data. The raw data has to stay like it is. You could have your result in a specific folder, like a scratch folder. If you need space, you could think, I could remove everything here. Is not problematic. It could be like intermediate files when you do analysis and so on. And the README that helps to understand what is going on in that folder. So the code is more reproducibility about code is quite specific. It's for those that make development. So there is not so many person here at MIVEGEC, but you are quite a lot anyway to do some scripting. So the advice I could give is always make the code publicly available. That helps to, anyway, if you do publication later on, it will be asked by most of the people of the reviewers. Track your code. It's to have a timestamp when it has been modified, by who, why, when. So for that, we have Git, which is great. It should be used in any case. Code must be documented. It means not only comment within the code, but also outside the code with a nice README to explain how to run a script and so on. And also have a help command within when you write a script that help for a new user when they use it. There is plenty of tools that could be used to have a reproducible code. If you use a heuristic, and is often in R, but we see it a lot in AI, please seed them and mention what seeds you have used, otherwise it's not reproducible at all. While producing code, always, if you have time and if the project worth it, implement test. It could be unit test or more integration test. And also, if needed, and you have plenty of steps well defined, and it starts to be really complex what you are doing, things to use workflow manager system, and here is the most used in bioinformatics is Nextflow and SnakeMake. I will come back to them later on. So the environment, now how to be reproducible about the environment. What is the environment? Actually is your computer, and the idea is to not mess up your computer. You should not end up with something like that. So it's more to think like tools have a lot of libraries or dependencies. Is like one tool could have 100 of dependencies. So each tool has 100 of dependencies, and a lot of dependencies are common to many tools. So it's a mess like that. But if you touch one dependency, actually it could touch a lot of other tools without you could see it. So to give a simple example, if you do an old school installation of a tool, let's think the tool A that have two dependencies. If you do an OS update or just install a new tool, the dependence B could have changed with a new version of it. What could happen with that? It could affect that the tool A may provide different results, or the tool A may stop to work and be broken. And even in that type of installation, if you need two versions of the same tool to do different things, it will not be possible. So you will have to use execution environment boxes. To make it simple, what are these boxes? It could be environment using like Conda. Conda is a tool that makes boxes for tool A, a box for tool B. And what is great with this kind of tool is like, even if the dependencies are already on your computer, it installs them in the box for the tool you ask for. So it will not mess up the dependencies with your computer. And it will not mess up neither dependencies between two different tools. And you could have tool A version 1, tool A version B, tool A version 3. You could have the same tool at different versions in parallel. So Conda is great because it's agnostic about the tool you want to install on your computer. Could be Perl, Python, C, you don't care. You just ask for the tool and they are already packaged ready for you to be used. There is a nice community that provide tools, or we call them packages. It's called BioConda or CondaForge. And there is more than 30,000 of packages that are freely available for you. You could catch like that. Just one command. But there is a drawback. Conda is not perfect for reproducibility because it's not completely hermetic. And it comes already compiled, the tool, so you save time when to get them. And depending if you are on Windows or Linux, maybe the tool are not available. It could be available on one platform, but not to another one. So it's why I promote the use as much as possible, the use of container. Container, what is that? A container is a standard unit of software that package a base image, like a mini OS, with all dependencies and the software you want to use. So the OS is behind because now we don't care about the machine where it's running the container. It will work in the same way on the Windows, on the Linux, or on the Mac. Reproducibility here is kind of perfect, and the container are more or less hermetic. Could be open if you ask specifically for it. So it's not dependent to the OS, and it's really light. It's much better than a virtual machine because it's start and stop like that. It's really, really light, and it's easy to share, actually. So there is two main tools that are the most used, I would say, in bioinformatics, is Docker and Singularity, and the particularity of Singularity is more for HPC. So if you are iTrop platform, iTrop user, is Singularity that you will use there. Where to find the tool if I don't know how to write a container? Because writing a container is not necessarily easy, but it's the same. There is a community that take care of that for you. And there is plenty of registry where you could grab containers. They are ready to use. You just have to download them, and they are 100% reproducible. So don't hesitate to go there. It's just a one command line, wget or curl. I don't know what you use to download, and you could use the tool you want on your computer. So what do I need to do to have a reproducible environment? So use most as possible open source software. When you use software, list all of them and with a version, specific version. Use as long as you can, Conda, or there is a Pixie also, or container that will help you in reproducibility. And always add in the analysis folder instruction for reproducing the environment, what you have used, which container, where did you download it, and what version. Always version is important to mention. So there is plenty of tools to help on that. This is maybe the most important one. So they are again here to help you to make your life easy when you want to do analysis, at least for bioinformatician. But they are here to help for the reproducibility problem. So should I spend time to think about reproducibility? Why it's an extra layer and I will spend time on it? Actually, again, you will have benefit. Maybe at the beginning it's difficult, but later it starts to be something natural for you. So before a project, it would help you to structure and organize your work. It will encourage you to think about the scope and limitation of what you want to do. During a project, it makes it easy for you to rerun analysis. If you want to update a tool, it's easy. If you want to update your data, you can rerun, it's easy. You want to change parameter, you could rerun everything. If it's reproducible, it's easy. It will improve collaboration. People that see a tool reproducible will say, can I use that? I have some other data, can we try together and so on? And it speeds up manuscript writing, at least the methodology section, maybe not the whole thing, but that helps a lot. And after the project, it will help your future self and also the others that can quickly resume your work and check what you have done, and will definitely boost your visibility and impact the scientific community. So few slides about open science. Actually, it's really few slides. Why open science? What is open science? So actually, it's quite simple, the concept. You just make everything open. So everyone could have a look at it. So it's not just we think about open access of publication, but is actually is at every level and make the citizens or population have a look at what is going on in research project. So it's the raw idea is more transparency in science and accessible to everyone. Why making open science? I found this slide, I like it. Actually, is for focusing on these 4 main points: the equity, integrity, this is really important, have a higher impact, and trying to have a collaboration, so exchanging the knowledge. So we should maximize the scientific and societal impact of discoveries. This is the idea behind. Okay, this is a great idea, but how to do that? So just a quick focus on the French ambition about it. We had 2 different laws that talk about that, about open science, and 2 a national plan for open science. So the law from 2020 is interesting because it says open science become a mission for researcher or teacher researcher. So now we should, it's a mission, we should be open. And how to do that? So the plan helped to raise some money, give money to try to do something interesting. And I think the more concrete stuff is the creation of the research data group, the National Research Data Platform that I guess everyone knows. But the idea with this plan is to promote and generalize that open access of publication, and we should reach the 100% of publication in open access by 2030. So it's really soon. We started in 2017 with 41% only that are open. I don't know where we are now in 2025. I hope we're around 70%. Who knows? But the idea about all this plan and this open science is to structure sharing and opening up research data, open up and promote the source code produced by researchers, by bioinformaticians, and transforming practices to make open science the default principle. So what we talk about, the fair, the reproducibility, actually should become the default nowadays, or soon, the soonest as possible. So concretely in IRD, we have a plan also, and we have the Mission pour la Science Ouverte, MSO, that has been developed here at R&D to make application of these different plans. They are here to help on that side. And concrete creation is a data suite portal, which is a data portal. Again, this is to put data there that will be open. So the take-home message with these three main pillars is like open science promotes the dissemination and sharing of knowledge. The FAIR principle ensures that this knowledge is easily accessible and reusable. And the reproducibility here guarantees that this research are really reliable and transparent. So together, they create a fundamental framework for modern research that should be rigorous and accessible. So I hope I have convinced you, but you were maybe already convinced. Yes, I know that a lot of the things you already heard for everywhere, and is because we are in a time where everything's changed and we have to repeat and repeat that this become the default. So, but all these things is not really possible without any computing, at least the bioinformatic parts. We have the data, we have environment, but we need to compute everything. And a lot of research or computing is made on your home computer, on your desk, but there is HPCs that are here because there is more and more data and more complex analysis, and we need really huge computer. So we are really lucky in MIVEGEC because we have the iTrop platform, a really nice infrastructure with many bioinformaticians and one IT that take care of this platform. We have a quality system in that platform that make it available at any time. Every day it's working properly with many tools available there. We have a certification ISO 9001. And our aim is to have a working infrastructure. So we support it, that it make it working all the time. And we train users to know how to use it in a good way. So iTrop is also involved in a lot of network and research and development with at R&D level with EASY. People never heard about EASY, is informatique scientifique de l'IRD. So we do some training with them related to informatics. We have training with networking with the Southwind. We were doing training in bioinformatics. This year, we will do training in bioinformatics only with the iTrop platform outside of Southwind. And we have some link with IFB that help us sometimes to do some training too. So this infrastructure is great. Just use it. Common is a big machine. We have more than 9 million CPU hours, and we use only 1/3 of it altogether with the other units. So there is plenty of space left there. So if you have data or any ID, there is plenty of data in public archive. Just grab them. You have an idea, a new pipeline, try it. Go ahead. There is plenty of persons that know how to use it, or you could get help, or you could come to me. Use that cluster. Is a big and great cluster. We are not so many users compared to the size of it. When you see outside how it goes, we are really, really lucky to have this cluster. It's really a chance. And the chance is we have a lot of money coming also from the IFB. And this year we will have 2 GPU server, completely new, and last version that we can do a great research with that. And even a new SuperMem server that will be 2 TB of RAM. And actually is more than 80,000 euros that will be spent to improve the cluster over the 2 next years. So this is really a great machine. Go ahead, use it. And the best way to use it is to use pipelines. So automation is good. Automation is quite common in bioinformatics, and because there is more and more complex things to do, so there's plenty of steps. And it allows you to standardize analysis. So you could write pipeline in any language you want, but the best is to use a workflow manager. A workflow manager system, what is that? Is a system that allows you to have your pipeline that is portable. You could use it, deploy it everywhere with ease. It's scalable. It could work locally on your computer, could work on the HPC, and the day where we don't have HPC anymore, you could even work on the cloud, on Amazon or Google Cloud, whatsoever. It optimizes computation resource, will do the parallelization for you, as we have hundreds of steps. This helps a lot. And improves the reproducibility. So the two main framework like that used in bioinformatics is SnakeMake and Nextflow. So if you see some SnakeMake or Nextflow pipeline somewhere on the net and you want to try it, do it. It's not so difficult, and we can help you. So I wanted to finish with 2 slides to say this is some example of pipeline that has been used this year by MIVEGEC users. So I know that a lot of you are aware of them, and different departments use different pipelines. But something that I wanted to mention is like frogs. This is a pipeline within Galaxy. So Galaxy is also a framework that I didn't mention, but I could have put here. Is also something that's good for reproducibility. And on the right side, actually, all this pipeline here is a Nextflow pipeline. So this manager of workflow. And this one has been published and developed within MIVEGEC last year. It's going the wrong side. So why Nextflow? Why there is so many pipeline Nextflow? And because it exists something is called the NF core, which is again a community. You remember a community about container, about Conda, and so on. It exists a community for pipeline, for Nextflow pipeline, and we do an effort to collect and create the best analysis pipeline using the best practices and the best standard. So it's more than 100 pipelines ready to use, fully reproducible. Go ahead, pick the one you want, see if you can put your data there, and you can do a lot of things. A lot of interesting things. So about pipeline in MIVEGEC, actually there is ongoing research and development. I wanted just to mention that. This is maybe something that people don't talk too much, but they are involved in different things to improve already existing pipeline, or to publish new pipeline, or to, yes. This one I said was published last year. This one I published it this year. It's a pipeline to do alignment. If you have reads, you want to align. I made the simplest tool ever to align reads. Just, you can call it in one command line, you do everything for you. So there is a lot of nice projects, and I know, but I'm not sure where it is. There is few SnakeMake projects around also, people developing SnakeMake. This is great, but one is mature. Don't hesitate to share it, and I hope in the future we could have a page to see what has been done at MIVEGEC about all pipeline and what is available and why. He's here. Yes, so it's the end. Thank you. So I've been fast because now we could have a kind of fun moment with a woo clap. So if I succeed to set it up, but maybe if you have questions, you can do that in parallel. Let's try. If you're ready. So first question, do you know the FAIR acronym? So it's not written in English here, but anyway, guess which one is the best. I will try to, I forgot to, I will not vote myself. So as you see, indirect the answer of the others, you could just adapt you. So yeah. A lot of respondents is already great. To have like 50% is great. No, it's not. It's 30, 35, 38. Okay. Were you sleeping during my presentation? At least the correct one is the one that has the most of results. The trap is reproducible, but that's good anyway to be reproducible. So let's go to the next question. Nobody for that one? Interchangeable was not. But why only the French is displayed? I'm disturbed. Should I minimize? Comment je rapetis ça? Ah ben oui, mais on voit plus rien. Vous voyez l'anglais dessus? Ah vous? Bon, ok. So for you, how important is that FAIR principle in your research daily work? So someone was not here during the presentation, maybe. So this is anonymous, so don't worry if the answer is wrong. But okay, I see a lot of answer for high importance. This is good. It has to be important. This person, we have to talk together to. I will convince him or her why you should try to put that more central in his work. Okay, let's go for the next question. It's time now for you to write. In one word, how do you describe your experience when you apply this FAIR principle in your research project? In French or English is fine. Ah, yes, like a diesel. This is true. Okay, I think we are reaching our. Ouais. So more people say, I like this answer, bigger it is. Yeah, you could like many answers. Not reproducible? So, right, this is difficult and challenging, and so let's say the practice is quite new, is only from 2016. I will say that is really something that is like 10 years. It takes time. I see like a diesel, yes, it takes time to use that as a default usage. There is a lot of loss. Okay. So everyone sees that as a challenging, and even for those that use it quite often is still challenging. There is always a lot of things to remember, and is really un travail à part entière nowadays. And it is why I think in a department like the size of MIVEGEC, it would be good to have a data manager, because this is something that he will deal with you, guide you, give you guide and help every day what has to be done from the production of data until the end it ends up in a database. But the data management plan is here too. That is not becoming challenging, but slightly easier, that the step is less high than it was in the past. So let's see in many years how it goes, if it's better or not. We goes to the next question, is not the last. Yes, how many of your publications are in open source, do you think, roughly? Don't go to Google to count on. because I remember you that we should reach the 100%. The thing is, most of you started in the past before it become such central, so maybe all papers are not open. So I think we are in the good range compared to what is expected. So we will be here in 2030. Good. Continue your efforts. We are going in a good direction. That's great. Need to see who is that. to see how it goes and how to. There is. Yeah, the thing is, often it could be expensive to have published in open paper, but there is more and more good papers that are really with a charge really low. So we have to be aware of them, and maybe we should give some tips to these 2%, or even maybe them. Je ne connaissais pas. Oui, le HAL. Le problème de l'embargo, ça, si tu n'as pas écrit, tu étais accès libre, ça n'avait pas vu. En fait, la bonne pratique, même si on ne publie pas dans du open source, c'est avant la version finale, mettre la version non finale. Dans ça, vous avez le droit dans HAL ou dans PubArchive ou PubArchive, BioArchive et MedArchive ou the Archive you want.
Parce que le petit problème avec HAL, c'est que ça a été conçu par un psychopathe. Franchement, c'est juste super chiant.
L'idée derrière est louable.
Oui, mais il y a BioArchive. Non, sauf quand ça ne marche pas.
Hein?
Quand ça marche, c'est super, quand ça marche pas, c'est unique. Moi aussi.
Moi, je ne mets jamais rien dans Raphaël, j'écris à une personne. Bon, tout. Bon, apparemment, on a des tips à se donner. Ça serait bien d'avoir une petite discussion sur le sujet. Tu veux dire quelque chose?
Articles were more cited than others, of course. So if you do not care about open science, but metrics, go ahead to open science. And also, I just checked, it's between 60 and 67% in
25, 24, okay.
In France, open science publishing.
So I think we are following the trend. Is good.
Can
Yeah.
I just add to that, not to compromise the quality. Sorry, thank you. Can I just add to that, not to compromise the quality of where you published? There's a lot of predatory publishers out there that make your articles publicly available. They usually charge a whole lot of amount, and there's a very little ethics behind that. And I say this mostly to the young generations. I'm something like an old lady, which I am, but this can actually, if you publish with certain predatory journals, this will harm your chances of getting a position, a permanent position. It can harm. And I'm talking very precisely about, for example, MDPI journals. Okay, this will actually not look good on your CV.
If you don't
so don't compromise the quality. There are some really good open access like PCI, etc., that you can actually rely on. Just yeah, be careful.
I don't disagree with
This is a big MDPI publisher. Just joking.
if you don't know which journal to choose, there is a nice database that has been developed here in Montpellier by Nicolas Galtier that lists all the journal, and it is a
Daphne is
or
called,
not,
yeah.
etc.
Yeah, so go to the Daphne website. If you type Daphne ISM, you're going to see a list of like sort of like ethical journals in all of the areas of ecology and evolution. You can choose a criteria called health, and that has a lot of the journals that are to do with any type of health, human health, plant health, animal health, etc. So I really encourage you to have a look at that.
So let's go to the next question that is close to B5. So if you would like to have some data from your colleagues, what strategy you will use, or you think it's worth it to use to access to her or his data? So it's always nice to offer coffee every morning. I don't know if it works to get data, but you could always start by that. Maybe this is not really mandatory, because by default in research, we know that if people use work or data from others, we are supposed to cite each other. So this is maybe by itself. I don't know, it's always good to remind it. But yes, transparency is good for everyone. I don't know who are your colleagues. You are unlucky for that person. So we can go to the next question. Did you get any difficulty when you try to reproduce analysis, or are you maybe for bioinformatician you see a tool and you want to try it? But for researcher, it's more like you see nice research, I say, I will try to reproduce it.
Sorry, I don't understand this question.
Tu vois des résultats, tu essayes de les reproduire. Ah non, là, oui, je l'ai fait en 2 fois, pardon. Celui-là, c'était propre résultat. Donc, tu as des analyses que tu as fait, mais tu dis, ah, il y a un truc qui me chiffonne, j'aimerais bien les refaire. Vérifier. Est-ce que ça se passe comme ça? Tu arrives à refaire, tu as toujours la même chose, ou quand tu... Côté expérience, c'est plus difficile, mais ça peut compter aussi, mais il faut être plus... Ouais. Je vais faire un zoom out pour voir mieux, mais supplémentaire. Comment on fait ça? Ah, pas mal. Ok. C'est marrant qu'il n'y a personne qui est... Tout le monde a déjà essayé de reproduire ses propres résultats, c'est bien. C'est que vous êtes...
C'était pas clair si tu parlais de reproduire l'analyse.
Tu peux... Les deux, ça peut marcher aussi, mais là, c'est vrai que je suis plus dans l'optique donnée, mais si tu réponds dans l'optique, ça... Mais au niveau manip, c'est bien plus difficile d'avoir quelque chose de reproductible. Bon, on a quand même des difficultés. Peut-être que d'appliquer certaines choses que je vous ai citées plus tôt dans le talk peuvent aider à faire face à ça. La question d'après, c'est la même chose, mais par rapport à des recherches de quelqu'un d'autre. Donc, vous voyez quelque chose, vous avez envie de vous l'approprier ou de vérifier le travail d'un collègue. Est-ce que ça marche toujours ou c'est très difficile? Les mêmes questions ont été posées à plus de 1500 scientifiques en 2016, c'était un papier dans Nature. Et il y avait la moitié des gens qui disaient qu'ils ne pouvaient pas reproduire leurs recherches. Donc, c'est ce que nous, on voit. Nous, c'était presque même plus que la moitié avait des difficultés. Et tout confondu. Médecine, computational, tout. C'est un peu uneven, mais il y a des difficultés partout. Et là, pour cette question, pour d'autres chercheurs, ça allait même à 75%. De personnes qui disent qu'ils sont incapables de reproduire les recherches de quelqu'un. Donc ici, on voit que, ben oui, on a quand même souvent des soucis. Il y en a qui sont chanceux et que ça se passe plutôt bien. Ils ne sont pas beaucoup les chanceux. Ouais, ben, il a une bonne source d'utilisation. Il y en a qui n'ont jamais essayé. C'est vrai qu'on n'a pas forcément le temps. Oui. Bon. Peut-être pas besoin d'essayer, et on sait que c'est reproductible. Si vous connaissez bien les gens, vous pouvez avoir plus ou moins confiance sur certains travaux. Là, je pense qu'on va sur la fin. À votre avis, il reste 2 questions. Quelles seraient les meilleures pratiques pour s'assurer que des travaux de recherche ne soient pas reproductibles? Qu'est-ce qu'il ne faut pas faire? Qu'est-ce qu'il faut faire pour que ça ne soit pas reproductible? Trafiquer les données, ça c'est très bien. Que des licences pourries, donc qu'est-ce qu'il y avait? Payantes, pardon. Oui, ça c'est bien aussi. Sans que ça vous amène en prison, j'aurais dû mettre... Ah, rien sauvegarder, oui, ça c'est parfait, c'est très très bien. Une seule copie dans un endroit qui craint, quand ça crache, pouf. Alors inventer des données, c'est pas si facile, mais oui, ça peut être une bonne chose. Ouais, virer les outliers, ça passe inaperçu, c'est très bien. Boire en travaillant, alors je déconseille fortement. Alors, document nothing, malheureusement, on le voit beaucoup. Mettre des espaces dans les titres de fichiers, c'est bien. Quelqu'un a suivi ce que j'ai dit. Ne pas utiliser GitHub, ne rien annoter, pas partager les codes. Ah, il y en a d'autres qui arrivent. Data et code en PDF. Alors, le code en PDF, ça, c'est magnifique. Ouais. Ça, c'est parfait. Pas de rides, pas de chocolat, pas de rides, je ne sais pas ce que ça veut dire. Tu te débrouilles. Ah, plot en PPT. C'est possible. Ah oui, non, on les crée carrément de toutes pièces. C'est technique, ça prend du temps. Je crois qu'on peut s'arrêter là, mais il y a des très bonnes idées. Il y a de très très bonnes idées. Donc j'espère que vous avez à peu près tout lu, vous voyez tous ces trucs un peu à éviter. Parce que la prochaine question, maintenant, c'est parmi tout ce que vous avez pu voir juste avant, est-ce qu'il y a des choses auxquelles vous vous identifiez et que vous faites? C'est pour ça l'inspiration que ne venez pas de nulle part. Il y a des mauvaises pratiques que vous mettez en application. Il va falloir essayer de les sortir, de plus les utiliser. Ah oui, non, mais c'est parce que oui, le WooClap était censé. Je pensais le faire au début. Donc oui. Il y en a qui préfèrent ne pas répondre. Et je crois qu'on a terminé. Il y en a une dernière. Ah oui, sur la science ouverte. Oui. Pourquoi? Quel est l'obstacle pour la science ouverte dans votre équipe? Donc, pensez à HAL. Pourquoi vous ne mettez pas vos publications dans HAL? Alors, les données top secrètes, ça se comprend. Ça se retient un certain temps, mais une fois que vous avez fait les papiers nécessaires, c'est bien d'aller de l'avant et de quand même finir à les mettre dans des bases de données publiques. Il y a 4 personnes qui sont super contentes de leur équipe, c'est bien. De quelle partie? Oui, c'est ce que je... Bien sûr. Ah ouais? Non, mais tout à fait, ça dépend beaucoup des disciplines. Ici, c'est une idée générale de chez nous, à MIVEGEC. On a déjà pas mal de disciplines différentes. Je ne sais pas quelle est cette équipe, mais elle mérite une médaille. Enfin, si ça se trouve, c'en est plusieurs. Effectivement, l'accompagnement, ça peut être aidé avec, je parlais de data manager, l'accompagnement peut être à plusieurs niveaux. Manque d'information, il faut trouver l'information, mais elle est là. C'est un peu la jungle, mais il y a de l'information, mais il faut la trouver. Pas de peur de critique, ça c'est bien. Et la paresse, oui, souvent, on a déjà beaucoup de choses à penser, donc c'est difficile d'appliquer tout ça. Bon, merci pour tout. Bon week-end. Bonne...